Thank you very much for inviting me to share a little bit of what's happened over the last, say, 100 years in, when it comes to hereditary colorectal cancer, where we started and where we are right now, and what the challenges are looking at the future. I do have no conflicts of interest to disclose. So the story starts in 1913, so over 100 years ago, with Dr. Warthen here. He was a pathologist in Michigan, and he described the family of his seamstress, which is right here, and he said that there were a lot of individuals with endometrial, particularly endometrial and gastric cancer, some colon cancers too, and he called it a familial cancer group. 50 years later, Dr. Lynch right here gathered together 600 descendants of this family, and he classified this as a truly autosomal dominant cancer syndrome that later on would be called Lynch Syndrome. 1925, just a little bit later, Dr. Lockhart Mummery from St. Mark's in London described 3 families where he said there were adenomatosis of the large bowel, and he said that those were they tended to develop it in succeeding generations. He was finding that these individuals would develop cancer, and they would develop colon cancer very early in life. So he really was describing familial adenomatous polyposis. About 100 years later, 1986, actually, Herrera and colleagues took on a patient who followed exactly this phenotype, which is multiple polyps, as you can see on the right side, as opposed to the left side where we see a normal colon. And they saw that actually these individuals had loss of, deletion of 5q. And that set the work in motion for finally in 1991 to end up describing APC as the gene responsible for familial adenomatous polyposis. So right now we do know quite a bit more besides APC, which was the first gene related to colorectal cancer syndrome. Now we know about the genes responsible for Lynch syndrome, for MAP, or MYH-associated polyposis, Peutz-Jeghers, juvenile polyposis, Cowden syndrome. So we do have a good knowledge right now on what the genetic defects are responsible for some of these colorectal cancer syndromes. And we've learned even more of that. We've learned that actually some of the genetic defects have a very clear implication on the particular phenotype of these individuals. For instance, this is the exon 15 from the APC gene, where we do have the mutated cluster region here, where 60% of the common mutations are. And for instance, individuals who have Gardner syndrome, which is APC with osteomas and soft tissue growth, do have mutations in this particular region of the mutated cluster region. Or for instance, attenuated forms of polyposis do tend to have mutations in the 5' region. So actually, it's not we not only know what the genes are, but actually we're starting to understand better what the relationship between phenotype and genotype is. The inheritance pattern of colorectal cancer are in all cases except one autosomal dominant, as far as we know right now. FAP, attenuated forms, Lynch syndrome, hamartomatous polyposis are all autosomal dominant. That means that we have one mutated allele that we inherit, and with that we are inheriting the syndrome. 50% chances of being inherited by descendants. There's only one syndrome that we know of right now that's autosomal recessive colon cancer, MAP, and so in this one we'll need to inherit 2 mutated alleles in order to have inherit the syndrome. Therefore, it will be 25% chances of inheriting disease. And as in most cancer syndromes, those do follow the 2-hit hypothesis of Knots, and basically we inherit a mutated gene from either mom or dad, and it is at the somatic level that we see the inactivation, mostly the result of loss of heterozygosity of the other allele, and therefore the development of the disease. When we look at the big pie of colon cancer, and we start mapping where all these syndromes fall into, the majority are still 65-85% will be sporadic, we have a big chunk also that we call familial cases, about 15 to 20%, depending on the series we're looking at. And there's been over 30 variants that have been associated with colon cancer, but actually only one, this one here in HQ, that's been particularly associated with familial cases. But again, those are variants, and those are just slight increased risk of developing cancer. And then we have these better characterized, well-characterized syndromes with still some gaps that we'll point out to, that we can divide between polyposis and non-polyposis. The polyposis ones are the ones that, like the one we showed you before, so phenotypically we can certainly diagnose, and we've been diagnosing them for years because of that phenotype of lots of polyps and other cancers and other problems that they have. The non-polyposis cases have always been the most challenging ones when it comes to diagnosis, and that's because we don't have that phenotype, the pre-morbid phenotype of lots of polyps, and individuals do develop cancer, and most of the time we just don't think that they can belong to these families. Lynch syndrome falls into this, but as we'll show you in a few minutes, probably there's a little bit more to that than Lynch syndrome in this particular group. And this is the group that's the most important group of the hereditary cases. So let's focus a little bit on Lynch syndrome. This is, as we said, autosomal dominant, penetrance 70 to 80%. Only a few polyps. Again, that's one of the challenges. Mean age at diagnosis depends on the series, mid-50s on average. Predominant right side of the colon, while sporadic cancers are 70% on the left side of the colon. Very rapid cancer development. If sporadic cancers, we believe, takes 8 to 10 years to develop into cancer, from small polyps to cancer, those can develop within 1 to 2 years, so very, very rapid development. And very common development of subsequent colorectal cancers. Up to 30% of cases will develop in 10 years, another new diagnosis of cancer. And indeed, what they are is that these are multi-cancer syndromes. That's the majority of colon cancer syndromes, as we can see here, comparing the risk of cancer for Lynch syndrome individuals or for sporadic cases. It's the risk of colorectal cancer is very high, but also we have endometrial cancer, gastric, pancreatic, ovarian cancer up to 10%. So those are multi-cancer syndromes, and that's how we should be approaching them. And we've also learned that the genes implicated do play a role in how the phenotype will behave, and also sex will have a lot to do with it. For instance, in terms of colon cancer, men with the same type of Lynch syndrome causing mutations will have a higher risk of colorectal cancer, but of course women do have that added burden of endometrial and ovarian cancer. On the other hand, there are two of the genes implicated or responsible for Lynch syndrome, MLH1 and MSH2, that they seem to be associated with a higher risk of cancer and a lower age at diagnosis. Therefore, they seem to be appearing earlier and more frequent in comparison to MSH6, for instance, or PMS2 that's not here. So the gene does matter. That happens at the same level with endometrial cancers too. So we're not only looking at if there is diagnosis of Lynch syndrome, but what genes are affected, because they do play a role. And actually, we are tailoring surveillance and recommendations according to the gene that's implicated, of the 5 genes that we know cause Lynch syndrome. So let's go back. So how do we make the diagnosis? This is one of the most undiagnosed syndromes, and that's something that needs attention, and that's always been that challenge, as we said before, because of this lack of this pre-morbid phenotype. Well, part of a substantial part of the armamentarium on how we make a diagnosis for Lynch syndrome is how the cancer develops in these tumors. When we look at the causes of genomic instability in colorectal cancer, the majority of colorectal cancers develop through this chromosomal instability pathway, the SYN pathway, where there is generalized loss of heterozygosity, and which is truly characterized by large aberrations in the karyotype and aneuploidy. On the other hand, when we look at tumors from Lynch syndrome individuals, those do not have these large aberrations. The cells are diploid, so they are very different, but what they have is what we call microsatellite instability. Microsatellites are these repeats that we find in DNA. They are spread throughout the entire genome, and in these cases, what we see is deletions or insertions in these particular repeating sequences. as these repeats keep getting reproduced, we'll start seeing mutations in genes that control cell growth and death, and finally that will end up developing into cancer. And that is the mechanism that ensues in these cases. So basically what we have is, as we know, our DNA chains keep making mistakes as they keep reproducing. Some of them will be single base mispairs or insertion deletion loops. When that happens, the mismatch repair system is a group of proteins that what they do is some of them do identify those mutations, and some of them what they do is actually get recruited by these other ones, and they act in consortium in order to fix these particular mutations. In Lynch syndrome, what happens is that one of these 4 genes is mutated, therefore this results in lack of expression of the protein. There's a faulty mechanism of fixing these mismatches, finally resulting in the phenomenon that we call microsatellite instability. In the majority of cases, the mutated gene will have a lack of expression of the corresponding protein, and that's what we can see in the tumor. So we see lack of expression of that protein in the tumor as opposed to the non-involved tissue. Important and key to remember also is that about 10% of all sporadic colorectal cancers do follow the same mechanism, the same carcinogenic mechanism, which is through this microsatellite instability and because of the lack of mismatch repair. From what we've known so far is that, and what we believe so far is that in all cases where that happens at the sporadic level, it was always due to hypermethylation of the promoter region of MLH1. Only this particular gene, and these other genes would not be involved in sporadic cases. That's been what we've known so far. So when we would have microsatellite unstable tumors, the only job here will be distinguished between sporadic cases with hypermethylation of MLH1 or truly syndromic cases due to germline mutations in the mismatch repair genes. So that's the molecular basis that's part of our algorithm for diagnosis. But how we come to that part, well, we've been talking for the longest time about what kind of clinical criteria can help us determine which patients should really undergo this type of testing to see if they actually end up having Lynch syndrome. And initially there were those Amsterdam criteria described, but basically they were a very restrictive set of criteria. Later on, the Bethesda criteria that I'm showing here that are part of still of the guidelines nowadays, we're saying if any of the 5 conditions that we are showing here are fulfilled by any patient, then patients should undergo the algorithm that we'll show in a minute, including microsatellite instability testing of the tumor. So young individuals, so individuals who have colon cancer at age less than 50, or individuals who have more than 1 colon cancer either at the same time or later on, 1 colon cancer plus another cancer of the group of cancers that are commonly associated regardless of age. MSI histology, pathologists can tell us a lot about it because there are certain features in these tumors that are quite specific, like the tumor infiltrating lymphocytes, for instance. Those are suggestive, and therefore it's criteria for suspicion. And then some family history criteria, having just 1 first-degree relative diagnosed at an age less than 50, or having 2 or more first or second-degree relatives would put us into the path of the diagnosis. There are some limitations, though, and we know that about 28% of Lynch syndrome mutation carriers do not meet these criteria. On the other hand, the rest of them do. So it's so-so, but still some. It's got some usefulness in terms of trying to screen initially who may have Lynch syndrome. Other ideas have come about, such as prediction models. Some of them are online. For instance, that was developed at Harvard, the PREM 126, and some studies showed that the cost effectiveness of these tests would be around 5% cutoff to undergo germline genetic testing. And at the end of the day, though, it's all clinical criteria that we just often don't think about. Therefore, we're still missing a lot of cases. So a push was started to say, well, let's actually do microsatellite instability testing or IHC of the mismatch repair proteins on all colon cancer tumors. There have been several studies that have shown the effectiveness of this approach, and actually in 2013 the NCCN guidelines started recommending this approach, and the Multi-Society Task Force last year did also recommend this, and that's actually what's been done at Yale for the last few years. So basically, universal testing, meaning every single colon cancer is tested for microsatellite instability or expression of these proteins. In some cases, another approach is just testing everyone who's younger than 70, and the reason to distinguish younger with older than 70 is that because most of the sporadic cases we know about, those ones due to hypermethylation of MLH1, are all individuals that in general are older individuals, so specificity does go down in those cases. But the approach that's taken by many and most institutions right now is universal testing. The key here is following up on all those cases who are actually really ruling out Lynch syndrome in those cases. So this is how we select. We can either select by clinical criteria, as we were saying before, or as we said, MSI testing directly or IHC in all individuals. As we mentioned, if there's loss of MLH1, it could still be sporadic tumor. Therefore, we can do BRAF mutation analysis, because about a third of these cases will have a particular BRAF mutation, but none of the Lynch cases do. And we can also look at MLH1 methylation, and again, only sporadic cases will have methylation. When we have loss of these other proteins, MSH2, MSH6, PMS2, we've thought so far that actually all these cases, none of them would be sporadic, all of them would have germline mutations, and so therefore we proceed to germline genetic testing. In a few subset of cases that we don't see loss of MSH2, we see loss of MSH2, but we don't find germline mutations. What happens here is that some of them are due to deletions in EpCAM, which actually results in hypermethylation and silencing of MSH2. But we've been noticing for years in clinical practice that many of those cases that in theory were Lynch syndrome tumors with loss of these three other proteins, actually we were not seeing germline mutations. And we've been treating them as if they had Lynch syndrome, but of course we could not screen family members, and we've been very unsure about exactly how to proceed with them. Erring on the conservative side, we've been treating them as Lynch syndrome. So we decided to look at what's happening to this particular group. That's with the two Spanish series that were consecutive cases, and we did the whole molecular testing with methylation of MLH1, with immunohistochemistry, MSI, and then germline testing. And we look at all those, 16 of them ended up having Lynch syndrome, but actually 43 of them did not, and they've been called Lynch-like syndrome. So those cases did have those loss of expression of those proteins that we talked about. They have microsatellite instability and all that, yet they do not have germline mutations in the mismatch repair genes. When we look at how they look, we see that these individuals are young, or they are definitely younger than the sporadic cases, and they predominantly seem to affect the right side of the colon as the Lynch syndrome group does. When we look at families from these individuals and we look at relative risk of cancer, we see that they do have an increased risk of cancer, these families do. It doesn't seem to be as high as Lynch syndrome. And they probably have a slightly increased risk of other or extracolonic malignancies. But we still don't know a whole lot about them. So are they Lynch syndrome? But mismatch repair mutations are technically difficult to detect with current tools, and we're just still not being able to detect those. Are they syndromic cases due to germline mutations that are different from mismatch repair genes, yet they result in mismatch repair deficient tumor? Or are these sporadic cases? So we started looking into that, and we used our Chicago series of 654 colorectal cancer patients from a multi-ethnic cohort that we had from there. And we proceeded with tumor testing for MSI and all of them, IHC, and BRAF mutations in all of them. Of those 654, 56 had microsatellite unstable tumors. And we look at the hypermethylation of MLH1. That turned out that 30 did have hypermethylation of MLH1. Therefore, they are those sporadic ones. 23 of them did have loss of any one of the mismatched repair proteins, none of them having BRAF mutations, none of them having methylation of MLH1. And out of these 23, in 18 cases, we've been able to look at them. And what we've done is we've done exome sequencing of both germline and somatic tumors. And to our surprise, actually, only about a half of those cases that potentially were Lynch syndrome, 23, only about half of them did have a germline mutation in mismatched repair gene. But in all other cases, what we saw is it was a double somatic hit. We saw in the majority of cases a mutation plus loss of heterozygosity, and in some cases 2 different mutations. So what's happening here? How unusual for a sporadic cancer to have 2 different types of mutations affecting the different gene and causing the inactivation of this gene. What's happening here? I think there's we do have a lot of interest in trying to figure it out, and that's what we're working on. So that's one of the questions that we still have up in the air, but we are really looking into. Another aspect, another one that we're looking into, and it's got a lot of interest, is these individuals who also look like Lynch syndrome. They do follow actually these very strict criteria, Amsterdam criteria that were described initially. That's basically what they show you is autosomal dominant pattern of disease with several members affected, with young individuals. So when we look at those, half of them are Lynch syndrome, half of them do not have actually mismatched repair deficiency, they don't have mismatched repair gene mutation. So what do they have? We certainly know very little about them. And we know that actually they are young also, not as young as the Lynch syndrome cases again, but they are young. But they seem to have more left-sided tumors, more in line with sporadic cases, and they definitely don't have lymphocytic infiltrating the tumors as Lynch syndrome, as many of the Lynch syndrome cases have. So what do they really have? Well, one of the aspects we thought it was important to consider is that the role of epigenetic events in the carcinogenic process of these tumors. So what we did is we looked at a group of tumors from these that we call hereditary non-polyposis colorectal cancer cases are MSS and not MSI, versus a group of Lynch syndrome tumors, versus another group of sporadic microsatellite stable tumors, and another one with microsatellite unstable sporadic tumors. And we look at CpG island methylation through 5 different markers, and we look at global methylation through the surrogate marker line 1 for global methylation. And what we saw is that in this group of MSS HNPCC, there were 0 high level CpG island methylation. And when we looked at line 1 methylation, that surrogate marker for global methylation, what we saw is that significantly this group had a much lower level of line 1 methylation, thus probably global methylation than the rest of the groups. So that was the first finding that really seems to put this group together. Yet we still don't know anything about it at a germline level, but there are some somatic features that seem to coalesce in this group. What that means? Well, global hypomethylation has been implicated in early steps of colon cancer development, and it does seem to lead to reactivation of otherwise silenced proto-oncogenes. It often precedes genomic instability in colon cancer, and it seems to affect stability of chromosomes, imprinting mechanisms, activation of normally silenced genes. So it seems that, again, this group, the carcinogenic process in this group seems to be different. How that's controlled, is there any germline mechanism that's driving this, is still yet to be known. So those are some of the challenges that we are facing, recognizing how much progress we've made and how many cases we're able to identify many of these families. I did not talk about surveillance, prophylactic measures, but certainly we do have those, and we know much more right now that now we've been able to identify syndromes just by the gene and not by clinical phenotypes. I want to acknowledge the people in my lab, particularly Rosa Chicola and all the collaborators in the US and abroad, and some of the funding institutions. Thank you.